Each cell within a muscle is called a fascicle. Each fascicle is comprised of several myofibers, mitochondria, and multinuclei. Each myofiber within the cell contains many myofibrils, which are contractile organelles of skeletal muscle. The sarcoplasmic reticulum encircles each myofibril, which releases calcium into the cytosol. The synaptic knob receives a signal from the neuron, opening the calcium channels, allowing calcium to flow in. Calcium pushes the synaptic vesicles, which house acetylcholine, to fuse with the membrane wall, which releases acetylcholine into the synaptic cleft via exocytosis. Acetylcholine is dispersed throughout the synaptic cleft and connects to acetylcholine receptors along the sarcolemma. It is required that two ACH molecules attach to each acetylcholine receptor along the motor end plate in order to allow the release of sodium into the cell. This then allows potassium to diffuse out. This in turn causes the polarity of the sarcolemma to jump to a positive charge as sodium is released into the cell. It then slowly decreases back to a negative charge as the potassium diffuses out. This rapid fluctuation causes an action potential to be created. The action potential is carried down the T-tubules until it reaches the sarcoplasmic reticulum. When the action potential reaches the sarcoplasmic reticulum, it triggers a release of calcium from the terminal cisternae. The calcium leaves the sarcoplasmic reticulum and travels down its concentration gradient into the cytosol. The calcium then binds to the troponin and tropomyosin along the thin filaments. The calcium attachment causes the troponin and tropomyosin to shift its position, exposing the active site on the thin filament. It also prepares the site for binding with the myosin heads. The myosin head, however, requires ATP to cause the head to extend into the high energy position. The energy from the ATP is used to bind the head to the active site on the thin filament, forming a cross bridge between actin and myosin. The energy from the ATP is then transferred into ADP and a phosphate. The myosin head releases the ADP and a phosphate, which causes the head to flex and bend to the low energy position, pulling the thin filament with it. The myosin head stays attached until a new ATP is attached to it, releasing it from the actin and repeating the whole process. The process will continue to repeat until signals are no longer received by the neuromuscular junction. When this occurs, the active transport pump from the sarcoplasmic reticulum pumps calcium from the cytosol back into the terminal cisternae. Calcium then binds with a chemical called calsequestrin and stores it until the fiber is stimulated again. After contraction is complete, an enzyme called acetylcholinesterase attaches to acetylcholine, breaking their bond. This in turn causes the choline and acetate molecules to return to the synaptic knob until the knob is further stimulated.